Today we will be using our blue physical science review packet. Go ahead and turn to the second page, which is titled Scientific Inquiry Review. Think back to the beginning of the school year. Do you remember doing the Kool-Aid lab? What do you remember about the scientific method from the Kool-Aid lab? Do you remember what the scientific method is? What is the scientific method? It is a specific way that a scientist solves a problem. Our problem was creating the best tasting Kool-Aid. In order to create that best tasting Kool-Aid, do you remember what step number 1 was? Step number 1 is what drives every single experiment that scientists ever do. It is simply asking a question. Questions can be like what, or how, or why, or when. And you take those questions and you focus them and you create an investigation. All questions come from careful observations from the world around us. Once we have a question, we can then do step 2. Step 2 is to form a hypothesis. Remember that a hypothesis is a clear statement about 1, what your problem is, and 2, what you expect the outcome to be. Hypotheses are written in an if-then statement. So for our Kool-Aid lab, you wrote something like, if I add 1 gram of sugar to 0.5 grams of Kool-Aid mix and 100 milliliters of water, then I will have the best tasting Kool-Aid. Now, once you have your hypothesis written, this is when things get exciting, right? Because then you get to do step 3. And step 3 is testing your hypothesis. Now remember, in a scientific laboratory, when you are testing a hypothesis, you must have a controlled experiment. And in order to have a controlled experiment, you must only be changing 1 variable at a time. Now, in your note-taking packet, underneath step 3, testing a hypothesis, you will see the question in italics, why do we only change 1 factor or variable at a time? I would like you to answer this question at this time. Let's talk about this term variable. Variable is any factor that is different from one group to another. So, think now to controlled variables. What is a controlled variable? If you said that a controlled variable is a variable in an experiment that is held constant from the independent variable in the experiment, you would be correct. Now, you can think about our Kool-Aid lab and all of the controlled variables that we had, or attempted to have. It could be the type of water, the temperature of the water, the amount of time you stirred your Kool-Aid, sugar, and water together. It could be the tasting cups that you used. It could be the brand of sugar we used, to the type of sugar we used, to the flavor of Kool-Aid we used. All of those things we attempted to control in our Kool-Aid lab experiment. Now let's look at 2 other terms. Let's look at the terms independent or manipulated variables and dependent or responding variables. Another way to look at the definitions of independent variable and dependent variable is to think of other scientific investigations going on out there, like cancer research. In cancer research, the independent variable, or what's being manipulated in the experiment, is the introduction of a cancer drug. This is the one thing that is being changed by the person conducting the experiment. So please write in your note-taking packet that the independent variable, or the manipulated variable, is the variable that is changed by the person conducting the experiment. That would then mean that the responding variable, or the dependent variable, is the outcome, right? So when a person has cancer and they begin taking chemotherapy, the manipulated variable is the chemotherapy, and ideally, the responding variable would be a reduction in cancer in their body. So remember that the dependent variable, or the responding variable, is the variable within the experiment that is affected by the change. So it's the outcome. If you want to look back to your Kool-Aid lab, our manipulated variable was the amount of sugar that we used in each trial. That is what we changed. And the responding variable would be the outcome of changing the amount of sugar, right? Which was the taste of your Kool-Aid. Make sure you define responding variable in your note-taking packet too. When you're done, go ahead and turn the sheet of paper over. You will see step 4 is now listed. Step 4 is analyzing results. What does analyzing results look like to you? If you said it's examining all information that has occurred in the experiment thus far, any type of observations or data you collected, you'd be correct. It is at this time that we undergo what's called a scientific critique. So we look at the strengths of our experiment and the weaknesses of our experiment, and we begin to do drawing conclusions, which is step 5. But notice, there's 2 different types of observations we can make before we draw conclusions. Let's look at those right now. We can have qualitative or quantitative observations. Let's talk about the word qualitative observations. Remember, in the word qualitative, you can see the beginning of the word quality. Do you see the beginning of the word quality there? When you make a quality analyzation, what you're really doing is you're using your 5 senses, right? You're talking about what you can smell or hear or taste or touch or see. So these are things that are not really measured, right? And this is different than quantitative observations. Quantitative observations, look at the word. Do you see the word quantity inside of quantitative observations? And when you think of quantity, you're thinking of things you can count. And quantitative observations are observations that deal with numbers. It's measurements. So think about things that you can chart or things that you can graph. Think about things that you can measure. These are quantitative observations. Please make sure that you record the definitions for both qualitative and quantitative observations in your note-taking packet. And now let's go back to that step 5 that I had begun to talk about earlier. Step 5, after looking at both your qualitative and your quantitative observations, determining the scientific strengths and weaknesses of your experiment, which we know to be called a scientific critique, we then move into step 5, drawing conclusions. Drawing conclusions is now determining, did your results match our hypothesis? If the answer is yes, well, then you want to retest your experiment. You want to verify that the results you received are correct. And if you answered no, well, that's okay too. Then what you want to do is, again, you want to retest. You want to check your procedures, make sure that there was no errors. If there weren't any errors and your hypothesis still did not match your results, then you need to discredit your hypothesis and create a new one. Remember that in all scientific experiments, we want to do repeated trials. Notice in your note-taking sheet, you need to define repeated trials. Repeated trials is the number of times that procedures are repeated. In all scientific investigations, you should try to repeat the experiment 3 to 5 times in order to achieve a most accurate result. And once all these things are done, it is now time to move to step 6. Step 6 in the scientific method is communicating your results. This can be done in a lot of different ways. If you look in the image here, you can use scientific journals, you can use things like radio, TV, news articles, brochures, or the internet. Now, at the bottom of your sheet, notice it says practice. There are 2 questions in which you need to read the sentence. Based off of the sentence being read, you need to determine what is the testable question in this experiment. Then you need to determine Is this experiment using qualitative or quantitative observations? Please make sure that you answer both questions 1 and 2 prior to class beginning tomorrow. Tomorrow, we will be conducting our own experiments based off of questions you have about your own life.